dear little champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to do a complete revision for biotechnology principles and processes, just like a one short video. Think of that friend of yours who gives a full revision to you during the day of exam. I used to go, I was not that friend, okay. I used to go and just listen to them talk, because it was a really nice thing, because everything gets refreshed. We would be able to recall things better. So it would be just like that, and I'm assuming you have watched the previous videos, children, okay. The number of questions that have come from this chapter is 5 in the previous year, so we can expect something around that only this year also. It can go up, it can go a bit low, but not too much low. Anyways, 4 questions and all we can expect, 4 to 5 questions we can expect each year, okay. Now 5 questions means 5 into 4, 20 marks for this chapter. Now that's something huge, which is why I thought I should be making a revision video also. In this video you'll sum up everything, okay, that we have studied. Chalo. So the basics of genetic engineering, what all do you know before you start off the chapter? First thing, there is a host cell. What do you mean by host cell? For example, there is a E. coli bacteria, okay, there's a E. coli bacteria, and this E. coli bacteria, you know, it has its own DNA. It will have its own DNA, and this will be the circular DNA. DNA, you know, DNA has segments known as genes, which would be coding for proteins. So this is the DNA which codes for protein and all the things. So I call it chromosomal DNA. It's the major DNA that they have. Now apart from this, they also have certain extra chromosomal DNA. They also have extra DNA, which is basically not coding for any proteins. It's just there, and they will replicate whenever normal replication happens. Now this extra chromosomal thingy that you see over here, the extra chromosomal DNA that you see over here, you have a name for it. You call them plasmid. Now why do I call this as a host cell? For example, if something is hijacked, we say that these are our hosts. Host means basically I am going to use their machinery for something else, for my benefit. So basically this is the host, and I am going to use them by taking out their plasmid, that is the DNA, okay. Or probably I'll make like this, it will, okay. So I'm going to take out their DNA, okay, and put my gene of interest. So this is the DNA that I have, and over here I have this segment of the DNA which is known as gene, and this is my gene of interest which I want to put over here. So what can be done, children? With the help of certain enzymes that are known as restriction enzymes, I can cleave the DNA. I can cut the DNA. So over here I am going to use restriction enzymes, and I am going to cleave the DNA, and I am going to put my gene of interest over here. Okay, done. Now you might be thinking, why are we doing this, ma'am? Basically, after we have put this gene of interest over here, I am going to put it back in the E. coli's body. The plasmid, I am going to put it back in the bacteria's body. So over here, this is carrying my gene of interest, so I call this particular plasmid as a vector. What do I call it? I call it as a vector because it is carrying my gene of interest. So vectors are things that carry something. For example, mosquito is a vector of plasmodium. Mosquito has the plasmodium thing and it carries to us. So that is a vector. Mosquito is a vector. Similarly, over here, the plasmid is carrying the DNA, the gene of interest, which is why this will be known as a vector. So I'm transferring this vector back again into the E. coli. Now what will happen, children? Once I put it back, now what will happen? I'll wait for replication to happen. So I said the chromosomal DNA, they will replicate when you have to give rise to 2 more E. coli. Similarly, all these things will also replicate. Replicate means to make a copy of it, right? So one more thing, to make a copy of something, they should be having a site which is known as origin of replication. What do you call it? Origin of replication. So in the plasmid there will be a sequence of DNA which will be known as origin of replication, or I call it ORI site. Okay, so this plasmid, if it does not contain ORI site, the enzyme which is responsible for replication would not be able to do replication. So ORI is very much important over here. Okay, so what I did, children, over here I have the host cell. I took the plasmid out, Okay, anyways, it is not going to hamper any of the things because it is not coding for any proteins. Now I took the plasmid out, I cut it with the help of restriction enzymes over here. Similarly, with the same restriction enzyme, I am going to cut my gene of interest also, and now I am going to take it and put it over here. Okay, with the help of an enzyme, okay, I'll tell you that also. With the help of an enzyme, these guys, this DNA, that is gene of interest, and the plasmid DNA, they come together. That enzyme's name is DNA ligase. That enzyme's name is DNA ligase. It helps in ligating things, making things come together. Then after I have implanted over here, I call this as a vector because it is going to carry my gene of interest back into the cell. Now once it is back into the cell, I have to make sure that replication is happening. How can I make sure that? Only if it has a proper ORI site, that is origin of replication site, then only the next enzyme, that is DNA polymerase, will come and attach over here and do the replication and give me multiple copies of my gene of interest. Okay, this is my gene of interest, it will make multiple copies and then I can take the gene of interest. So this is the basics of genetic engineering, my children. Over here I am engineering the molecular thing. Okay, I am going within the cell and I am engineering stuff. Okay, so I hope you got this. We studied this only. Then we spoke about the first recombinant DNA. So it was found by Stanley Cohen and Herbert Boyer in the year 1972. About Stanley Cohen, I think it's given in front of the biotechnology introduction chapter also. And okay, so basically you have to know about all these things. It's very important. So Stanley Cohen and Herbert Boyer in the year 1972 saw the first recombinant DNA. How did they find it out? Basically, there was a bacteria whose name was Salmonella typhimurium. You know? Are you aware of Salmonella typhimurium? Well, this causes the typhoid disease. What does it cause, children? It causes typhoid disease. Okay, so Salmonella typhimurium again is a bacteria. It also has a plasmid. Okay, it also has a plasmid. Now they were under this procedure that like, okay, we need to find out certain things. We need to talk about certain antibiotics which could like they were in this quest of finding out things. Okay, so Salmonella typhimurium, for suppose I am putting ampicillin on Salmonella typhimurium, it will die because ampicillin is an antibiotic. Okay, antibiotic as in it will kill the bacteria, right? But now just imagine that Salmonella has a resistance gene for it, has an antibiotic resistant gene for it. Then what will happen? As in, if I am Salmonella typhimurium and I have a gene in me that is antibiotic resistant gene, that means I am having ampicillin resistant gene, for example. I am having this gene probably, that means that even if you put ampicillin on me, I will be able to survive. So this was the idea. And how did they come to the conclusion that this can be done or not? Obviously, I cannot change the sequence in the Salmonella typhimurium and put it there, because in that case, I am creating a type of bacteria, a Salmonella typhimurium, which will be resistant to any sort of antibiotic. So I do not want to do the procedure in them, but I want one thing from them. And what is that one thing, children? I want the plasmid from them. So I took the plasmid from them. Okay, then I put my gene of interest into the plasmid. Suppose this is the plasmid that I got from them and this is the gene of interest that I have. Now I cannot put this plasmid back into Salmonella typhimurium because I am putting what, children? Antibiotic resistant gene. So it will be resistant to antibiotic. Now if I cannot create a variety of Salmonella typhimurium which is resistant to antibiotic, then all our medicines will go in vain. So we don't want that. Instead, what can I do? Instead, I can do one thing. Basically, I want to check whether it is getting copied or not. That's the thing. No. So this plasmid, which is a recombinant plasmid, okay, I am going to put it in another bacteria, that is E. coli. I'll put it in another bacteria, that is E. coli, and check whether the replication is happening or not. And I found that replication is happening. So the same thing can happen here also. So this is how the first recombinant DNA was found. So the question comes, okay, that what organism was used for this, that is Salmonella typhimurium, but plasmid was taken from them and it was inserted in E. coli. Now what was the gene of interest, children? It was having antibiotic resistant gene. Okay, that was a gene of interest. Now which year? 1972. Cool? Works? I hope you got it. Now apart from this also, there is something that is written over here. We'll just have a quick read and then we'll go ahead. Okay, so Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistant gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotics. And yeah, so basically I also took it from another plasmid. I took it from another plasmid, okay. this antibiotic gene, I took it from some other plasmid. Now, the cutting of DNA at specific location became possible with the discovery of the so-called molecular scissors or restriction enzymes. So, cutting of the DNA, obviously, I have to cut this particular portion and put it over here. Okay, here also I have to cut. So, basically, what do I need, children? I need restriction enzymes. Restriction enzymes, you call them as molecular scissors as well. What do you call them, children? You call them also as molecular scissors. Cool? Works because they are cutting the molecules, that is DNA molecules, right? Now, the cut piece of DNA was then linked with the plasmid DNA. This plasmid DNA act as vector. Agree? They are acting as a vector because it is carrying the gene of interest. Now, to transfer the piece of DNA attached. You probably know that mosquito acts as an insect vector to transfer the malarial parasite into the human body. In the same way, a plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. Now, going ahead. The linking of antibiotic resistant gene with the plasmid vector became possible with the enzyme DNA ligase. Only when DNA ligase is there, they will join the two different strands of DNA there. Okay, now this makes a new combination of circular autonomously replicating DNA created in vitro and is known as recombinant DNA. What do you call this DNA? Where I have the old plasmid, okay, I have the old plasmid and I am putting my gene of interest over here. What do I call this? Plasmid, okay. So, what do I call this as? Recombinant. Now, this is a recombinant plasmid or recombinant DNA, my dear children, because it is recombined to form something new. Okay. Now, when this DNA is transferred to Escherichia coli, a bacterium closely related to Salmonella, it could replicate using the new host DNA polymerase enzyme and make multiple copies. The ability to multiply copy of antibiotic resistant gene in E. coli was called cloning. And what do you call this process as, where you are making multiple copies of thing? You call this as cloning. And basically, you are cloning a gene specifically. So, I call this as gene cloning as well. What do I call this? I call this as gene cloning as well. So, as of now, we studied about the basics of genetic engineering. We studied about the first recombinant DNA that we had, and now we go ahead with the isolation. Now, we are going to do the whole procedure. Okay. So, for that, the first thing that we need is DNA. First thing that we need is DNA. We have to isolate the DNA, either if it's gene of interest or probably from a bacteria. I need plasmid, whatever it is. I initially need a DNA, right? So, isolation of DNA is the first step. First step. Under that will be breaking the cell open. Okay, so you know DNA is inside the nucleus in the case of eukaryotes. In the case of prokaryotes, it's freely lying there in the nucleoid region. So anyways, whatever cell it is, it will be having a covering outside. So for example, if this is a plant cell has a covering outside which is known as cell wall, and cell wall is made up of cellulose. Okay, now the next thing is fungi. Fungi again has a cell wall, and the cell wall is made up of chitin. Okay. Now, the third thing, it's important, okay, this piece of information is very much important. Bacteria also have cell wall, and it is made up of peptidoglycan. So, children, we need to break the cell walls. Obviously, we need to break them. So, what all do we need? We need enzymes for that. So, the first enzyme for cellulose breaking, we need cellulase. So, you would be asked to match the correct enzyme. You should be knowing it. For chitin, you can use chitinase. For bacteria, it's the most easy thing. You can use lysozyme. So, in our tears, we have lysozyme. In our saliva, we have lysozyme, which is why it is produced, because any bacterial attack it comes, lysozyme is produced and the cell wall, everything breaks off, not basically the bacteria, and it will die. That's what we want. But over here, little amount of lysozyme is used. We don't want to kill the bacteria, we just want to take the DNA out of it. Okay. So, that is one thing. That is a step 1. Remember the name of the enzymes that are there, questions have come. Next thing. So, when I try to take the DNA, along with the DNA there are chances that proteins could come, yeah. Along with that there are chances that carbohydrates could come, fats could come, RNA could come. So, I should be treating this solution with different types of enzymes. So, for proteins, for treating with proteins, I should be having an enzyme which is proteases, which would cleave the proteins off, Carbohydrates, for that I have so many things, amylases, so many things are there, okay, amylase, whatever there are different types of carbohydrates we can use for that. Now, for carbohydrate I am not mentioning because it's not there in your book. For lipids I will tell. For lipids, I have So fats comes under lipids, yeah. So I have an enzyme that is lipase, okay. For RNA, you have RNase. So you have these three enzymes. What are we doing, children? Along with the DNA, if the proteins are coming, we need to remove them. So for that we have proteases. For breaking down fat, we have lipases. For breaking down RNA, we have RNases. Then, so we broke the cell open and then we took the DNA out. Along with that, all these guys came, we killed them. And now the next thing that we are going to do is pooling. Now the next thing that we are going to do is pooling, where I have a thing, a test tube. Okay, I have a test tube in which I would be having the DNA. I will centrifuge it for sure, and then after that I will be putting something in it. I will be putting a solution in it which is very important, that is chilled ethanol, my dear children. What am I putting? Chilled ethanol I am putting, okay. And when I am putting chilled ethanol, what happens to the DNA, children? The DNA, it will get precipitated. It will get precipitated. So I am writing here DNA gets precipitated. You got it? So we broke up the cell, we killed all the other things, unwanted things, okay. Now we did spooling, where we are using chilled ethanol to put it in there so that DNA could get separated. And over here you can see with this, the help of a spoon, we are taking it out. Not exactly a spoon, but yeah, okay. So this has to be done. So you have to remember the name of the enzymes, children. They'll ask you the mismatched ones. This whole thing that I am saying is important, which is why I am saying 10,000 times it's important. Okay, so this step 1 is isolation of DNA. We did it. Now, after we have isolated, we have to separate the DNA fragments. Actually, before separation comes something else, that is basically cutting of the DNA fragments, which is done by restriction enzymes. So that should be the second point actually, but okay, okay. Let's study about this first. Okay, so over here, you see this is electrophoresis chamber. Okay, you call this procedure as gel electrophoresis. And what is so special about gel electrophoresis? Let me tell you. So these are the wells where I am going to put the DNA. After cutting them off, I am going to put it over here. And here I will be having an electrode which should be negatively charged, and here also I will be having an electrode which should be positively charged. Okay, this negatively charged one I call it cathode, positively charged one I call it anode. How do I remember it? Listen, cat has paws, so basically cations are positive. Cations are usually positive, but when it comes to electrodes, CAT stands for negative. The opposite of it happens. So cations are positive, but cathode is negative. That means anode has to be positive. Okay, either way, that's how I remember the charges. Now, I cut my DNA, right? I cut my DNA with the help of restriction enzymes, and I am putting in the well. Why I am doing all this thing? Because I want to separate them on the basis of their length and everything. Okay, and this is connected to a power supply. And over here, I am going to use a gel which is known as agarose gel. Children, you can assume whatever I am teaching you today gives you a question. You can assume like that, okay? So agarose gel is extracted from seaweed. Fine. So over here I have that, okay? Now I am putting my DNA. The DNA basically would be moving around. The larger ones would be staying back, okay? The smaller ones would be moving really fast, okay? The smaller ones are moving really fast. So one very important thing, why is it moving towards the anode? Because children, the DNA that you see over here is negatively charged. So these guys will repel from each other. It will move from the negative pole because they are going to repel, minus repelling goes to the positive side, that is the anode. So DNA is negatively charged, which is why it moves. That is the basis of movement. Now who moves first? Smaller fragments are first in category. Okay, they are first in category. Please remember that. Done. Now one more thing I will tell. In the gel electrophoresis chamber, the DNA is not moving on the surface. Please remember that. Okay? I am writing it again. DNA does not move on the surface of the gel. On the surface of the gel. Then where is it moving, children? The gel, no, that you have, it has a sweeping effect. Okay, so that means in swinging, how do you see things, right? Similarly, the DNA also moves within. Okay, because, why? Because, children, gel has sieving effect. So the DNA will be moving from inside. Now, has question come from this, ma'am? Yes, the question has come from this, which is why I am saying. So they asked you to find the incorrect statement. So if you are not aware about this thing, you would mark it correct only. Okay, so they said that DNA is moving from the surface of the gel electrophoresis chamber. No, it's wrong. Now, the DNA is here. Why did I do all these things? Because I wanted the smaller fragments there, the gene of interest, which was small in size, right? Can I see the DNA over here? No. It is not something that would be visible. So what can I do, ma'am? Basically, I take a sheet, a nitrocellulose sheet, I put it on it, and now I take a sheet back, and I see that DNA is on the nitrocellulose sheet. Okay? Now I have everything on my nitrocellulose sheet. Now what will I do, children? Suppose this is a nitrocellulose sheet. I have these smaller fragments over here, larger fragments somewhere over here, whatever, just like how it was there. Okay? Now, still it is not visible, so I treat it with, this is again very much important, that was chilled ethanol which I was putting for spooling. Over here I am putting ethidium bromide. What I am putting, children? I am putting ethidium bromide, which is a stain. Okay, now this goes and enters into the DNA bonds and stuff like that, and that's how it works. Now can you see it, ma'am? No, still now I cannot see it. Next thing that I need is again something very important. To see it, I need UV. Okay, I need UV. Yes, UV radiation. Now I will be able to see it. At this point of time, this would be appearing bright orange in color. Please don't go write any other color. It will be bright orange in color. Now I can view things properly. So this is how separation of DNA fragments is important. What type of questions could come? They could ask you what is used for viewing things. Ethidium bromide. At what light? Infrared light? Normal visible light? It is UV radiation. What color does it give? Bright orange color, not red, orange. Okay. So in the well of the gel, this is the well, okay? In the well of the gel, is the DNA put towards the negative electrode or the positive electrode? They can say that it is put near the positive electrode. So that is wrong. It would be the negative electrode. That's when it will repel and move towards that side. Get it? So all these pieces of information, children, is very, 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 very important. And please remember, it shows sewing effect. Done? Next. You will thank me later, I am pretty sure about that. So, now discovery of restriction enzymes. The next thing, as I said, we had to cut it before, but okay, we will talk about it now. How were restriction enzymes created? So, people again, they were doing a lot of processes, things like the experiments, and now they had Escherichia coli, and this happened in 1963. That was done, the first recombinant DNA was found in 1972. This is done in 1963, where what has happened, children? Escherichia coli is there. It's a bacteria, okay? And this poor Escherichia coli is usually infected. Suppose this is Escherichia coli. Okay, it has the circular DNA. Okay, now I have a virus which I call as bacteriophage. This is the bacteriophage. Bacteriophage will be having its own DNA or genetic material, whatever is it. This guy goes and infects this guy. How? I'll tell you. So basically, it will pass down its genetic material and leave everything outside. Now this genetic material, as you can see over here, will go and get incorporated over here. So suppose this is the DNA strand, and over here I am going to incorporate the genetic material of this guy. Now obviously This is the gene of interest for it, right? So it will prepare its own proteins from this. Whenever the normal DNA undergoes replication, obviously this part of the DNA will also undergo replication. It will make its own proteins, and now you will be having chunumunu bacteriophages which will be going out. Okay, so that's how things happen usually. But then it was noted that basically this is twist in the story, and the twist was that the Escherichia coli which is there, okay, suppose this is the DNA and this is the genetic material of the bacteriophage. It was found that Escherichia coli was releasing restriction enzymes over there. Now these restriction enzymes, I told you, each restriction enzyme will be cutting at one particular site. So there will be multiple sites for different enzymes. So many enzymes, they have so many sites also. So anyways, the restriction enzyme which was being produced, it was cutting the DNA. Now you have small fragments of the DNA. Will it work now? No. So one more problem is there. If it is producing restriction enzyme and the site, okay, restriction enzymes work by understanding the site, and this is known as recognition site. So if a DNA has the recognition site, it will go and cleave that particular portion of it. So they have recognition sites and it is getting cleaved. Okay, the foreign DNA is getting cleaved. Now to save themselves also from this, they release another enzyme which is known as methylase, my dear children. What is that? Methylase. It will add methyl group. This methylase enzyme helps in adding methyl group over here and thereby saving from the attack of the restriction enzymes that they themselves are producing. So this is how the first restriction enzyme was thought to be seen or thought to be discovered. So I'll tell once more, bacteriophage usually passes out its genetic material. It goes and gets incorporated into the genetic material of the bacteria, and then when normal DNA replication and everything happens, the amount of copy number also increases and the proteins and everything is being made. That's how things usually work. But over here things are quite different. Okay, in the case of Escherichia coli, what happens, children? It is producing restriction enzymes. This restriction enzyme will have multiple recognition sites over there and will cleave each of them. Okay, now to protect themselves from this, they have also released methylase. They also release methylase, children. So it had methyl groups over here so that nothing could happen to their own DNA. This was done 1963, and the first restriction enzyme which was found was HIND2. Can you remember this? This is HIND2. This is the restriction enzyme that was found. Cool. Now it was found that HIND2 always cut DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. So its recognition site consists of 6 base pairs. Please remember it. 6 base pairs are there. 6 base pairs, as in, for example, there is another enzyme which has a restriction site like this, GAATTC. Over here I have again 6 base pairs. Similarly, this HIND2 also have a recognition site of 6 base pair. So they will have specific sequence and it will be universal. Everywhere HIND2 will be cutting that particular sequence only. Okay, now besides HIND2, today we know more than 900 restriction enzymes, children, and these restriction enzymes have been isolated from over 230 strains of bacteria. So 230 strains of bacteria was responsible for giving out 900 restriction enzymes. We never know what questions could come from this. Until now, question has not come from this para. But as I said, we never know. HIND2, it is important, the first restriction enzyme that we have and the whole story. So 900 restriction enzymes from 230 strains of bacteria. Remember it? Now, the next thing is naming of restriction enzyme. How do you name it? So, I have a bacteria, Escherichia coli RY13. With the help of this, I name the enzyme, that is EcoR1. This is a restriction enzyme. This thing that you see over here, it is a restriction enzyme. And how is it named? So, I take the first letter of the genus name, okay, E. Then I take the 2 letters of the species name. Then I take the first letter of the strain. This is RY13 is a strain, okay. RY13, it is the strain. Now 1, because in the order of discovery I put like 1, 2, 3. So this is in the order of discovery. So question has come from this simple thing also. What does CO stand for? CO stands for coli, Escherichia coli. Now I said that each enzyme has its own site and you call this as recognition site. What do you call it as? Recognition site. So recognition site of E. coli is this GAA-TTC. Now which direction to which direction? 5' to 3' direction. 5' to 3' direction. I say that this particular sequence that you are seeing, it is palindromic. I say it is palindromic. What do you mean by palindromic sequence? When I read from both the sides, it sounds the same. So over here GAA-TTC, don't read CCTT, no, not like that. GAA-TTC, and then again 5' to 3', it is GAA-TTC. So either way, from 5' to 3' if you see, both of them are sounding same. So basically it is a palindromic sequence that you see over here. Works? Okay. Please remember the code, the sequence of ECO R1. This is very much, very important. Okay, so I am writing it 10,000 times. They have asked the correct sequence. So 5' to 3', so G has 5 side. Okay, please remember that. Now, going ahead, the working of the restriction enzymes. How do you think that they work? So, again, there are certain points over here which is very much important, but before getting into the points, I want to explain certain things, okay? How do they actually work? So, see for it very carefully. So, I have the DNA, children. This is the DNA strand, where this is 5', this is 3', this is 3', and this is 5'. Cool? Works? Here I have GAA-TTC. So here I will be having CTTAAG. Okay. Similarly, 5' to 3', I have GAA-TTC, and here I will be having CTTAAG. Aren't they complementary? They are complementary. Now this is a part of a DNA. You have more to it, but I want this particular sequence only. So I am going to use what enzyme, children? I am going to use my ECO R1. I am going to use ECO R1. And ECO R1, as I said, this is the sequence that they have. Now in this sequence, they will make a cut between G and A. This is fixed for them. This is fixed. In the recognition site that they have, they will make a cut between G and A. So they are going to make a cut over here. Here they are going to make a cut like this. Here they are going to make a cut like this. Here they are going to make a cut like this. Now, how will the DNA appear to me after the cutting and everything has happened? So, over here the DNA might look something like this. Here. Where I have AATTC, okay, and here I have G, then this whole DNA strand, right? Here again I have G, and then C, T, T, A, A. Now what is this side? This is 5' end, this is also the 5' end. So I took my gene of interest and I see that they do not have the complementary base pairs over here. Similarly here also I do not have complementary base pairs. That's a problem. So these guys can stick to anything that becomes out to be complementary to them. So I call this as sticky ends. What do I call it? Sticky ends. Now the twist of the story is that I am going to use the same enzyme. I am going to use the same enzyme to cut the plasmid where I am going to put this DNA. So suppose this was the plasmid, my dear children. Ayyo, wait. This was the plasmid that I had, which was having GAATTC over here. This is 3' and here it was having CTTAAG over here. Now similarly, I am going to make a cut over here. So this will get cut over here and this will get cut over here. What do I get after getting it cut? If I am making it cut like this, what do I get afterwards? This area will be gone. Okay, wait. I'll show you how it will look, okay? I'll show you how it will look and that will give you an idea of things. So, over here, I'm making the smaller version of it. I will be having G, just G over there, and the inner side which will be there, it will be having okay, CTTAA over here. Okay, this is one thing. The next thing that I will be having is, I will be having a G over here, this G, agree? And there will be the whole strand over here. This will be the whole strand over here with what, children? Here also it will be AA, TT and C. Cool? AA, TT, C. Fine. Now what will happen? This guy will come and attach over here. This thing that you see will attach over here. So what do I get? Look at this very carefully. Okay. So what do I get? Here I have a G. Okay. Here I have a G of this particular strand, and I have AA, TT, C over here. I have AA, TT, C over there, and G at the end. So I got G. I do not have space to make all the other things. So there is G also over here. Now similarly here I have this thing. So again G will be there at the end. I will be having G at the end. Wait. Did I miss something? G, A, T, T, C. Okay. I should have made it a bit larger, no? That's why this confusion is happening. Wait. I don't want to confuse you guys. How can I make this? Back. Okay, we can make it here. Okay, so what do I have over here? I will do one thing. I'll pause the video, make it, and then I'll show you. Okay, just give me a second. Okay, so listen to me carefully. Fine. So over here, if you see, I had the sequence. I have separated G from AATTC. So AATTC is that side, G is this side. Similarly here also, I have separated G that side and AATTC over here. So in the picture, the next image that you will get, you have G at one end and AATTC at another end. Let's see that. So you have G at one end and AATTC at another end. Similarly, G at one end and AATTC at another end. So you see they do not have any complementary base pairs. I have made it. That's of the gene of interest. But still, actually speaking, they do not have any complementary base pair. Similarly, here also they do not have any complementary base pair. Now over here, if I put this there, if I put this in between, what will happen? These guys which are there over here will get a complementary base pair with this, will form a complementary base pair with this. Okay, because I have an extra sticky end over here, this will go and bind with this. Okay, suppose I have A, T, T, A, T, T, and then there is one C also. Let me put that C over here. I'll get G from there. I'll get G because G was already there. G is already there over here, so it's okay. Fine. So this end is clear. Over here, I made a cut like this where I had G at one end and AA, TT. Oh, was it? Just a second. Not AA. Oh, this is actually very confusing for children. AA, TT thing over here. So this was the second strand that I was having. Okay. Similarly, in the next strand that will come coiled around like this, I will be having one sticky end over here and the other one will be just normal G. So over here, this is the sticky end that I have. This will form complementary base pair to it. So here I will be having AA, TT, and G was already there. So I can include C also there. So this is how the sticky ends they work. The complementary sticky ends are very much important, my dear children. So eco R1 has a restriction site like this where it cleaves between G and A. Now remember that this is a whole DNA. In between the DNA I am cutting it. So this type of restriction enzyme, I call this as restriction endonuclease. So the one that cuts within the DNA, endo as in within, okay. The one that cuts within the DNA is an endonuclease. One that cuts at the ends of the DNA is exonuclease. Okay, so you have two. One is endonuclease and one that cuts at the end is known as exonuclease. Now, where does a restriction enzyme cut? even if it's endo or exo. Where does it cut? So basically, if this is a palindromic sequence, this is a palindromic sequence, children, this is also a palindromic sequence. Where is it cutting? Is it cutting between this? No, it is cutting away from the center. So away from the center, it has cut over here. Away from the center, it has cut over here. Similarly, away from the center, it has cut over here. Away from the center, it has cut over here. So within a palindromic sequence, it is cutting towards away from the center, a bit away from the center. In a palindromic sequence, I am not talking about the DNA. In the DNA, if you are cutting it within, it is with the help of restriction endonucleases. Now I am just talking about a palindromic sequence. In a palindromic sequence, things are being cut towards one side, either side of the palindromic side, but not at the center. Okay, usually not at the center. It's like center also certain enzymes cut, but we don't have to study about that. So all this little information are very much important, which is why I am stressing onto it very much. So each restriction endonucleases functions by inspecting the length of DNA sequence. First thing that they do is inspect the whole length of DNA sequence, find their palindromic sequence. What happened to me, God? Okay, find the palindromic sequence. Now, cut each of the two strands of the double helix at specific points in their sugar phosphate backbone. So, you know the DNA basically which is there, it has sugar phosphate backbone. So, your restriction endonuclease which is going to cut, it is going to cut the sugar phosphate backbone. So, you know one thing, inspecting the whole length of DNA is one thing. Second thing is cutting at the sugar phosphate backbone. Third thing, each restriction endonuclease recognizes a specific palindromic nucleotide sequence. It will recognize a specific palindromic sequence. No two restriction enzyme will have same palindromic sequence. Okay, they will always have different restriction site or recognition site. Now, restriction enzymes cut the strand of DNA a little away from the center of the palindromic signs, but between the same two bases on the opposite strand. So over here also they cut between G and A, over here also they are going to cut between G and A itself. This leaves single stranded portions at the ends. You get single stranded portions at the end. What do you call them? You call them sticky ends, my dear children. Now these are the names because they form hydrogen bonds with their complementary cut counterparts. So obviously if you have complementary cut counterparts, what do? You make hydrogen bonds over there. Okay. Now, this stickiness of the ends facilitates the action of the enzyme DNA ligase. Now comes the entry of next enzyme, that is DNA ligase. So, there is a question that has been come. Sticky ends are being joined by DNA ligase, and in the NEET thing, they said it is wrong. So, sticky ends basically they make their own complementary base pairs, but when I have to join two separate different strands of DNA, that is over here, here. I need another enzyme which is known as DNA ligase. So, I am repeating what I said in your exam. It has been said that sticky ends of the DNA is not being attached with the help of DNA ligase. So, please note that even though partially it is true, I don't know why they have said that, but yes. So, according to them, it's just for joining two different gene sets or DNA. For that you use the DNA ligase. So, when cut by the same restriction enzyme, the resultant DNA fragments have the same kind of sticky ends, and these can be joined together end to end by using DNA ligase. Okay, that is what is exactly explained over here as well. Nothing else. So, that is the working of restriction enzyme. Once you cut with restriction enzyme, that's when you do gel electrophoresis, dear children. Okay, and then you separate along with the fragments. Now, you got the fragment, that is something that you wanted. Now, you amplify the DNA. You make copies of it, and you put these copies into different vectors later on. So, first thing, you isolated the DNA. Second thing what you did was cut them with restriction enzymes. Third thing that you did was run them in electrophoresis chamber. Okay, you got the smaller fragments, you take them, and now you are going to amplify it so that I can put it in multiple vectors. So, for DNA amplification, I have a machine which is known as PCR. What do you call this? Polymerase chain reaction. Now, this is the name of the process and also the machine. Over here, the first step that you have is known as denaturation. What do you call it? Denaturation. And denaturation occurs almost at 95 to 98 degrees Celsius, that high, 99 or sometime it goes. Okay, denaturation means you have the DNA strands, you are going to cut them open. What are you going to do, children? For example, this is the DNA strand that you are having. I am going to cleave all the bonds over here, and now I will be having two separate strands of DNA, one in 5' to 3', other in 3' to 5' direction. This process is known as denaturation. Second thing that I want to do is I want to make a copy of it, right? So for that I need to replicate. For replication, I need the help of an enzyme, that is DNA polymerase. But DNA polymerase says that I cannot just start doing things like that. I need a primer. I need what, children? A primer. So the next step will be, I am putting it over here, and I am going to 5', 3', 3', 5'. DNA replication takes place in which direction, children? From 5' to 3' direction. So if it takes from 5' to 3' direction, then it has to join over here. So this is 5'. Okay, from this direction it will go. Okay, and what do you call this strand? As leading strand. Anyways, so for starting off replication, I told it needs a primer initially. So first thing is binding of the primers like this. So here also there will be primers that are bound like this. What do you call this? These are primers, my dear children. Okay, and this process of putting primers over there, allocating over there, you call this as annealing. What do you call this? You call this as annealing. Okay, and what is the temperature for this required? 50 degrees Celsius, 50 to 60 degrees Celsius. But you see, from 98 you have come down a bit. Okay, now the next thing that you need to do is extend the DNA with the help of DNA polymerase. So over here again I have a strand, 5', 3', 3', 5', and I am making a copy of DNA over here. This is extension in 5' to 3' direction. Similarly over here, extending from 5' to 3' direction. This is known as extension, children. This is known as extension. This is the third step. So you will be asked about the order of things. So denaturation is the first thing, annealing is the second thing, and extension is the third thing. For extension you need almost around 72 degrees Celsius. Again you have increased the temperature. Now if you have noticed, you are basically increasing the temperature, and this is a very it's a process which has high thermal activity, right? So and you know that enzymes do not usually work at this particular temperature. So what is that enzyme which is used for replication over here? You know replication is only done by an enzyme which is known as DNA polymerase, but there is different varieties of it as well. Why not take DNA polymerase from an organism which is responsible for staying in very hot environment? So that particular organism, children, is Thermus aquaticus. It is what? Thermus aquaticus. Another important thing. Aquaticus. Okay, this particular bacteria stays in very hot environment. Now Thermus aquaticus, I take T from there and AQ from here. And now I have made another enzyme which is known as Taq polymerase. I didn't make it. So the enzyme which is extracted from Thermus aquaticus, okay, it is a DNA polymerase, but you have a specific name for it. I am writing it here again properly. It is Taq polymerase. It's DNA polymerase itself. It is responsible for replication, but just I wanted to name it specifically because it is got from Thermus aquaticus, which is why all these things are happening. Now one very important thing, I have put primers. Apart from primers, what do I need for extension, children? What do I need for extension? So you know there should be bases coming here, okay, and bases is a part of nucleotide, so you need nucleotides. So what do you call them? You need deoxynucleotides. You need multiple deoxynucleotides which could come here, make a whole thing over there. So DNA polymerase plus Taq polymerase plus deoxynucleotides are also required for the whole process to happen. And when you do this, you get thousands, millions of billions of copies. Okay, you get billions of copies with the help of just this simple procedure. So we have amplified the DNA as well. Now what? Now what's the next step? Chalo. Next step is to make a competent host. We want to make a competent host. What do you mean by competent host? A competent host is a one that is being prepared to take up the vector DNA. Okay, to take up the vector DNA, that is a competent host. And how can you make a host competent, not competent, okay, competent? For example, I want to make an E. coli competent. What can I do with it? Okay, first thing, the question that you should be asking to yourself is that I already taught, but still it's revision. Why do you think the DNA cannot get inside normally? There should be something. It's why it's a very huge task. Why am I discussing about it? Because children, remember, DNA is negatively charged, and when I say it's negatively charged, that means it is hydrophilic as well. It is water loving. It loves water like anything. It's a negatively charged thing, so it will attract water molecule and stuff like that. Okay. Now, you know, we all have certain membranes, and you have to pass these membranes to get inside. Now, our membrane is usually made up of phospholipids. And they kind of hate water. So phospholipids, if you see, it has a hydrophilic head, like it loves water, that head particular region, and there is a hydrophobic tail that hates water. So suppose this is a cell, so there will be water content inside, there will be water content outside, so I have to hide the tail region. So what do? I make an inverted layer like this. So the tail region which is there, it is inside. So core region is hydrophobic. The core region which is there, it is hydrophobic. It hates water, and the thing that loves water stays outside. Now, if I want to pass DNA molecules, obviously that is a big problem, because it's a negative thing. When it goes inside, it will attract water molecules, so water molecules can also come across it, and the whole lipid bilayer could get destroyed because of this thing. That is the actual problem, my dear children. Okay, that is why I cannot do this process in a very simple manner. I have to do certain other things. The first thing that I do is heat shock treatment, and this is done usually for bacteria and stuff like that. Okay, now what happens in heat shock treatment? First thing, I am going to treat them with divalent cations. Divalent cations as in those which has a valency of 2. Why do I treat with divalent cations? Because I am shielding the lipid layer and the DNA. Both are shielded, so their negative charge and everything won't be seen. They are shielded. Okay, now they can easily pass through. Now this will increase the efficiency with which DNA enters the bacterium through pores in its cell wall. So I said pores in its cell wall. So this makes it really easy for the bacteria to get in within the pores of the cell wall. How did we create this pores, children? What was the reason? How did we actually create the pores? First thing that you should be understanding is that recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA on ice. So first thing that I do is I treat it with ice. So I have the recombinant DNA and I also have ice. Okay, around it, this is the host that I am looking forward for. As of now, it does not have any holes. It's sealed. Okay, I am treating it with ice. Then I am treating it with heat. And at what temperature, children? 42 degrees Celsius. At 42 degrees Celsius, I am treating it. Now what will happen? This will create the problem because it was ice first, that is minus degree, then heat 42 degrees Celsius, then again I am putting it on ice. So this is first, this is second, and this again I am putting it on ice, which actually, when I am treating with heat only, that point so many pores are being created which will take in this particular DNA. And when I treat it with ice again for the third time, like second time, what will happen? It will close such that nothing will go out as well. This is known as heat shock treatment. Okay. Well, over here when there is pores, who help in shielding everything is the calcium ions, that is the divalent ions. You need divalent ions, then only things will happen for shielding things. Even though it's pore, still lots of things can happen. So to safeguard from it, you need cations, that is Ca2 plus or Mg2 plus things. Okay. What is the first thing that I am doing? I am treating with ice, then with heat 42 degrees Celsius, it creates pores, and now again I am treating with ice so that it gets sealed after making it enter. This is one step. So recombinant DNA can then be forced into such cells by incubating the cells with recombinant DNA in ice, followed by placing them briefly at 42 degrees Celsius and then putting them back on ice. This enables the bacteria to take up the recombinant DNA. It has taken up the recombinant DNA. Okay, this is for bacteria. Now, for plant and animal cell, things are quite different. Okay, you have gene gun for plants. You have gene gun for plants. And you have micro injection for animals. Okay. In the case of gene gun, first thing, gene gun is also known as biolistics. Please write it somewhere. Biolistics. And what exactly happens over here? I am going to take certain microparticles. I am going to take certain microparticles of what, children? Of gold and tungsten. This is very much important. Gold and tungsten. Now, after taking these microparticles, I am going to cover it with the DNA, not DNA like this. I am not putting thread over things like that. It's very proper procedure, okay? So anyways, I am going to cover it with DNA, and then I am going to put it inside what, children? For suppose this is the plant cell, I am going to put it inside the nucleus of the plant cell. What will happen if I put it in the cytoplasm? There will be another enzyme such as nucleases which will cut the nucleic acid. So that is a big problem for me. I do not want that. Okay. In this way I am passing this down. In the case of animals also same thing. Suppose this is an animal cell. I have the nucleus. With the help of a micropipette, small thing, I am making things inside. Okay, I am putting my DNA of interest inside. This is known as micro injection. How can you remember what is for what, children? Gene gun. Plants are really strong, so they can take up this. If someone put gun on us, probably we will die. So for us, little injection is fine. So animals, for them you have micro injection. For them you have gene gun. Fine, works, easy. Okay, now, okay, cool. Now this both things that you saw over here, it is vectorless. Okay, this method is vectorless method where you are not using vectors to make your DNA inside. Instead of that, you are using these many things. And the last method that you can use is disarmed pathogen vectors. So basically a pathogen which is going to cause a trouble, I am going to remove that portion of the pathogen which is going to cause a trouble, and I am going to put the gene of interest. We will talk about that really soon. Okay, I think the next thing that we are going to talk about is that only. So that is it. Now the type of colonial vectors, as I was talking about, okay, you have different vectors for different organisms. For bacteria, you have bacteriophage and plasmid. I told you about bacteriophage, how it infects, right? So bacteriophage, they put their DNA, and then suppose this is the bacteriophage which is there. Okay, it has its genetic material. What I am going to do is, anyways, this genetic material is going to get incorporated into the equalized genetic material. Okay, so why not cleave a portion of this and put my gene of interest there? So I am putting my gene of interest over here, okay, and then this is going to infect, and now I have my gene of interest over here as well. This is in the case of bacteria. So bacteriophage, which is a virus, is going to infect this. Now plasmid also can be used. You know plasmid, how it is used. So you have the plasmid, you take the plasmid out and you put the gene of interest that you want, and then you put it inside. That is one way. For bacteria, so these are the two ways for bacteria. For plant, it is Agrobacterium tumefaciens. This is very important. So this is a pathogen. It's a bacteria that causes disease which is known as crown gall disease. So what does this usually cause, children? It causes crown gall disease. This has come. Okay. So what is very unique about this is Agrobacterium tumefaciens, no, it has this plasmid, okay, which is known as TI plasmid. TI stands for tumor inducing plasmid. Okay, and TI plasmid is going to cause tumor by how? Basically it has a portion which is known as tDNA or transfer DNA. So what happens is that this plasmid gets out, okay, and gets inside the plant cell, So within the nucleus, so over here I have this thing with the tDNA. This tDNA that you see over here gets incorporated there with the plant's DNA, and it does its function. It gets incorporated there and it does its function. But now, what can we do with it? Is that, okay, now when it gets in there, obviously it will cause multiple cell division. And now you will be having so many cells with uncontrolled growth, and that will cause a cancerous sort of thing, okay, which is why now the disease is known as crown gall. It is known as crown gall. Now over here, if I remove the tDNA, and instead of the tDNA, if I am putting my gene of interest, which will be in blue color, so now this gene of interest will be going there and the plants will be producing that only. You get it? So instead of instead of gene of interest, I am writing is replacing what, children? The tDNA, and everything is fine then. Cool. Now the next thing is for animals. That's how things takes place in the case of plants. For plants we have this, okay. It's a very important thing, please do not forget. Now the next thing for animals, what do we have, children? Retroviruses we use. You know HIV is a retrovirus. Similarly, you have so many other retroviruses, not HIV obviously. We are not going to use that for doing cloning things in the case of animals. So these are the different cloning vectors. You should be knowing their names, very much important. Okay, so many questions have come around this and this and this. Okay, so just assume whatever I am teaching is important. That's what I said earlier also. Now features of a cloning vector. How much is left? Just let me see that. Okay, it's going to end. Features of a cloning vector. So this is a vector that we spoke about. What all things should be there in them? So in their whatever DNA replication is there, they should be having an ORI site for sure. They should be, for example, this is the plasmid I took. It should be having an ORI site because ORI is responsible for initiation of replication. It is responsible for initiation of replication. Children, apart from that, it is also responsible for copy numbers, as in how many number of copies can be created, that is also decided by the ORI. So the one that which has ORI that supports many copy number, you should be taking that. Okay, that is about ORI. Next is selectable marker. What do you mean by selectable marker? Basically, if again this is the plasmid, I should be having areas, for example like this. This is a gene which will be coding for ampicillin resistance. This is a gene that codes for tetracycline resistance and stuff like that. Why do I need it? So basically, suppose I have, instead of the tetracycline resistant gene, I am putting my gene of interest there. Now, when I am treating it with tetracycline, will this survive, children? Will this survive? No, it won't survive because it has my gene of interest, and I will be able to find out that this has my gene of interest. So this is acting as a selectable marker. This is going to act like a selectable marker. So these enzymes that you see, they are going to act like a selectable marker. So you should be having cloning vectors which has selectable markers in them, so that we would be able to understand which one has our DNA, which one does not have our DNA. Next thing, it should be having cloning sites. What do you mean by cloning sites, children? For example, this is the DNA. Now you know there should be sites, recognition sites for enzymes. So these sites, for suppose, these are the enzymes recognition site. So here cut will be made, and here whatever thing that I need to have is inserted. This is known as a cloning site. This is same as recognition site. So here only the cut and everything is being made, and this is a site where I can get my clone also. Every time this particular thing will replicate, what will I find, children? Here I will be finding my gene of interest as well. So this is the cloning site. So I am trying to actually say that, even though in your NCERT it is directly written as cloning site, I am actually wanting to say that there should be site for multiple enzymes. Okay, for example, EcoR1 is an enzyme, they should be having a site for that, or some other enzyme, BAMH1, it should be having a site for that. Suppose whatever enzyme we have, it should be able to cut with the help of that. So that should be, the number of sites should be like that. Okay, now I want to tell one very important thing. Okay, listen to me very carefully over here. So I am saying that this is a plasmid. This is a recognition site for EcoR1. Okay, I should be having just one recognition site for EcoR1. Only one for one restriction enzyme. There should be only one site for one restriction enzyme. Why, children? Because suppose EcoR1 has site over here, here, here. What will happen? Your whole plasmid will get cut. That's a big problem. Next thing, there should be different sites for different enzymes. or basically multiple sites. There should be multiple sites for different enzymes, as in one enzyme site is this, next enzyme should also have its site, next enzyme should also have its sites. You should be having multiple different sites. I'm not saying one enzyme should have multiple sites, no. Each of them should be having their own site over here so that I can use any enzyme over here. So that is one very important thing. I hope at least not by this statement, whatever I said, that is understandable to you. Okay. So one particular enzyme should have one site over here, but you should be having multiple enzymes also. For example, one is EcoR1 site, another one is BAMH1 site, another is SAL1 site. So you should be having sites like that. That's what I meant. Okay. Now the next thing that we have is two very important type of vectors. One is PBr322 and another is PUC8. Let's see about PBr322. Over here, this is the plasmid that we have. Okay. I don't know why I got tired, I think. Anyways. Okay. This gene is coding for ampicillin resistance. This gene is going to code for tetracycline resistance. As in, so this particular plasmid, if for example an organism that has this plasmid will be able to survive even if ampicillin is there, even if tetracycline is there, both of them are antibiotics. Cool. Now apart from this, they also have ORI site over here and ROP site over there. So ORI site is for origin of replication, and ROP is another protein that helps in initiation of replication. Okay. Now the thing is that in this gene only, in the tetracycline gene only, you have two sites. One is for BAMH1 and the other one is for SAL1. You have to remember the name of enzymes. So this is an enzyme which will make a cut over here. This is an enzyme which makes a cut over here. Okay. Now over here also you have two enzymes. Again, one is PST1, another one is PVU1. So recognition site for these enzymes are over here. Now you have one enzyme over here also. This is PVU2. Okay, 1, 2, 3, 4, 5 enzymes are done. Now you have three enzymes are over here, recognition sites, EcoR1, then you have CLA1, and then you have HIND3 children. You have HIND3. So this is 2, this is 3, rest everyone is 1, 1, 1, 1, 1. So you have how many enzymes? 1, 2, 3, 4, 5, 6, 7, 8. 8 enzymes are there in this particular PBr322. Now what is the use of this? Listen. So PBr322 is a plasmid that can be used as a vector for any bacteria and things like that. Okay. How can I use it? For suppose BAMH1 enzyme is being used. Okay, it will make a cut over here and I am going to put my gene of interest over here. So instead of tetracycline resistance, now what am I having, children? I have my gene of interest over here. I have put my gene of interest over here. Now I am going to do heat shock treatment, whatever is there, and I am going to put it in host. So there are three probability of hosts that I will be getting. Okay. One that will have this particular thing which has the ampicillin resistance, okay, which has the ampicillin resistance and the gene of interest. Another one, it is the normal thing, the normal plasmid, the non-recombinant. So this one that you see over here, this is a recombinant plasmid. This is a recombinant plasmid, this one. There will be a non-recombinant also where you will be having your tetracycline resistant gene over there. There will be a non-recombinant over here which will be having your tetracycline resistance over there. So this is the ampicillin resistance and the tetracycline resistant will be over here. This is the non-recombinant one. Okay, so what is happening basically? This is the non-recombinant one, this is the recombinant one, and over here I say transformation has not taken place. I do not know about it, but as of now. How will I come to know that whether transformation has taken place or not? What do you mean by transformation? When the vector DNA is gotten inside the host, you call this as transformation. You call this procedure as transformation. Okay, now over here I say transformation has not taken place. How can I come to know that? Basically, if I treat it with ampicillin, I will get to know that. How? Because this will be able to survive as it has ampicillin, this also will be able to survive as it has ampicillin, but this won't be able to survive. That means both of them, in this cases, transformation has taken place. These are both transformants. Both of them are transformants. Okay, I do not know whether recombination has taken place or not, whether they have the recombinant one or not. How will I get to know that, children? I have to treat it with tetracycline. But if I treat this with tetracycline, I will get to know one thing, that is basically this will be able to survive, which does not have the gene of interest, but the one that has the gene of interest would not be able to survive because it has lost the tetracycline ability. So I cannot kill it. So what do? I make a replica plate of it. And then I treat it with tetracycline. And I get to know that the one that is above is not able to survive. That means it has lost the tetracycline resistant gene. Instead of that, it has the gene of interest. But the one that is their lower side, I will arrange in a way I get to know what is what. Okay, I'll get to know this is the one that has tetracycline. That means recombination has not taken place over here. And I'll be collecting this one because it is not having tetracycline resistance. That means it has gene of interest. Now this process is known as insertional inactivation. This process is known as insertional inactivation. What do you mean by insertional inactivation, children? You are inserting your gene of interest, which is why it is losing its activation. Okay, inactivation, it is losing its activation. What is tetracycline resistant gene which was there? It is losing its activity because you are inserting the gene of interest there. It's about PBR 322. Now there is one more thing. Okay, there is one more thing that you should be knowing, that is about PUC8. Where can I make it now? Do I have something? Just a second. Okay, I will make it here if it's fine. Okay, I'll make it here. Listen, this is the next gene, next thing that I have is PUC8. Over here also you have ampicillin resistant gene, and the next thing that you have is another gene which is LAG-Z gene, children. It is LAG-Z gene, and LAG-Z gene, it codes for an enzyme that is known as beta galactosidase. Now this is also very important. So suppose there's a bacteria that has this plasmid, okay, which is coding for beta galactosidase. Now if I am putting a substance, a chromogenic substance over here, chromogenic as in when it breaks down it will give out color. I am putting a chromogenic substance that is X-gal. When I put it over here, this reacts with this, okay, reacts on this and basically gives a blue color. So I understand that yes, beta galactosidase is still being produced. Now what I am going to do is that I am going to remove this and put my gene of interest over here. Now if I put my gene of interest, do you think it will get activated? No, this will get inactivated. So will you get beta galactosidase? No beta galactosidase. So if beta galactosidase is not there, even if I put X-gal, do you think it will give me color? No. So basically I'll get colorless colony of bacteria. Colorless colony of bacteria, my dear children. So I can choose the colorless ones that say that they are the recombinants, because the recombinants has lost the ability, which is why they are colorless. If they didn't have recombination undergoing over there, then obviously they would have given the blue color. Got it? Cool? Works? Okay, now just give me a second. Okay, now the next thing. So we got the vector, we cloned it and everything happened. We have chosen the best vector which has the ORI site. We chose the vector that has a good selectable marker, good cloning site, or basically recognition site for different enzymes and everything. Okay, we spoke about PUC8. Did it in your NCERT? They didn't put this name, that is PUC8, but you should be knowing it. Okay, PUC8. Now the next thing is that I have to culture the bacteria that has the DNA, the plasmid, the vector, whatever it has. Okay, I have to, the gene of interest is there in this particular bacteria, and I want to culture it. What can I do for it? How do people, how we treat it? We are given food, we are given air, and that's how we grow. Similarly over here also, they need the basic things. For example, they need air. They need air, children. That's the most basic thing that they need. Okay. Second thing, they need nutrition. They need nutrition. They need something to mix them well. Because obviously if you are not mixing, what happens? There will be certain bacteria will be saying, yay, we got oxygen. But there will be other bacteria which will be saying, we didn't get oxygen. So that will be a sad scenario. We do not want that. And they will undergo something else which is known as anaerobic respiration. So do we want that, children? No. We want aerobic respiration. And mostly all the bioreactors, they will, all the culturing things will be having an effluent treatment also, where all the things are being removed from that particular thing. They will have a pH detection thing, they will have a foam detection thing, so that there is no too much foam being made over there inside. Okay, there will be a thermal jacket, lot of things. So these are the basic things that is needed. Okay, now we will go ahead and see how can you culture them. So there are two ways to culture. One is small scale culture and the second one is large scale culture. Small scale as in we do not have much requirements, little amount of it is required, the proteins or whatever is being made, it is very required in very less amount. So for that I have two methods. One is batch culture. Okay, one is batch culture and another is continuous culture. In batch culture what I am doing is, I have everything, I am giving air, and along with air I am giving nutrients also, but these nutrients are fixed nutrients. Okay, it is fixed nutrients, and to mix I also have the whole thing and everything there. Okay, so what happens is that the air and the fixed nutrients, the bacteria will be like so happy, they will all grow, okay. So initially if the graph will be like this, there is a lag phase, lag phase, and slowly it will increase. Now this is the lag phase or exponential phase. Why just a second, I am messing things up, I don't want to mess things up. Lag, log, exponential. Yes, so what I was saying previously is correct, I'll write properly. This is lag phase, then you have log phase. Okay, in log phase there's an exponential growth. Now everything is all cool, but after some time, since you are giving fixed nutrients, obviously do you think they will be able to survive more? No. So what will happen? There will be a plateau phase. They won't be increased. So I have to take them out and I have to put the next batch. That is what you mean by batch culture. Okay. Now there is next thing that you have is, it is known as continuous culture. Over here, what you are going to do is, you will be giving air, you will be giving nutrients, but this time unlimited nutrients, children. And over here you will be having an effluent tube, okay, which will be carrying out the product that we want along with the waste materials and everything. So you are giving everything and it is being removed also continuously, okay, and the new things are being added up continuously. Everything is a continuous thing that is happening. New things as in new nutrients, everything is being put, the bacteria is all same, they are dividing, you are taking it out, you are using again, same things. So over here the graph is kind of different. The lag phase is there, then there is log. Okay, that is the growth in them. So continuous culture is far better. Okay, you don't need to put too much attention to it, but yeah, you need to provide nutrients and air and everything in a continuous manner. Over here, it's like fixed nutrients and you take the next batch. So this is a small scale production of things. Now there is large scale production of things, and for that you need something that is known as bioreactors. Okay, you need something that is known as bioreactors, and in your NCERT textbook there are two bioreactors given. Okay, both of them comes in this category of stirred tank bioreactor. Okay, it is stirred tank bioreactor. What exactly happens over here? So I will be having a thing to stir the whole content. That is this thing. Okay, here also I will be having a whole thing to stir all the content. Why? Because now in your NCERT it is given that you want to make sure that there is availability availability of oxygen. Now you might think ma'am is saying something and I won't pay attention. It is very important. Questions have come. Why do you need stirred tank bioreactors? For availability of oxygen continuously. Now again you have two different types. One is simple stirred reactor that you can see over here. The other one is sparged. Okay, the other one is simple stirred reactor. Other one is sparged. What happens in sparged reactor? Over here, the thing that happens is there will be air bubbles, and air bubbles, as you know, they can easily go and diffuse, and it can give more better way of oxygen transport inside the bioreactors. So this would be the one that is more preferred in comparison to simple one. Okay, in comparison to simple one, we use sparged one, that is a better quality one. Okay, done. Now that is a basic thing, that is even nothing else. And now then the next step and the last step is, so first thing you isolated the DNA, then you cut it with restriction enzyme, then you made it run in gel electrophoresis chamber, then you amplified the DNA, then you put it in the vector which has a good ORI site, which has good selectable markers and everything, and then now you have cultured them. After culturing and everything is done, okay, when the expression of the gene happens, okay, when proteins are being made, I call this phase as biosynthetic stage. What do I call this as? Biosynthetic stage. This is when the proteins which are there that are being made. Okay, now I want the proteins. This is the product that I want actually. So the first step is I will separate the content. Okay, I will separate it from the waste products, from other unwanted stuff, and I will be having just my product, main product. Then I will purify it, then I will add preservatives to it, and then I will be doing a quality check. Well, these three processes, that is separation, purification and addition of preservatives, comes under a thing that is known as downstream processing. This is a question that has come in NEET. What all comes under downstream processing? Exclude the one that does not come. Okay, so separation, purification and addition of preservatives. You're separating things, you're purifying it completely, okay, and then you're adding preservatives. This comes under downstream processing. Quality check is the next thing, next step that we have, and this is the last step of your chapter as well. This is an important question, please remember it. I've been continuously talking, so that's why I'm a bit tired, but yes, this is what it is, and I hope that you got each and everything that I tried to explain, and with this we have completed the complete revision of this chapter, that is biotechnology principles and processes. I've told you the areas from where the questions can come. All the things that I remember and I'm teaching you are the areas of the questions. So please make a note of it, and tomorrow you have the mock test. Please attempt it once, because you will feel so happy after attempting the previous year questions, because you might be able to attempt all of them. And do let me know the marks as well, because I will be so happy to see you score really good and increase your confidence as a teacher. That's the biggest gift that you can give. Okay, so that's all, my dear children. I hope this was helpful. If it was helpful, definitely do not forget to subscribe to our channel, and do let me know in the comment section if you have any doubts, if you like my teaching, whatever is it. If you didn't like, then also. So yes, I will improve. Bye bye, take care.